The Eukimatoid Repository for Sustainable Industry and Livelihood Enabling Community Empowerment, or eResilience, is a project that envisions the continuous effort of expanding the culture database of novel and commercial strains of eukimatoids and economically important macroalgae. Since the seaweed farming success in 1970s, most of the commercially cultivated strains are more than 60 years old. Due to persistent clonal propagation of the crop, its overall productivity, carrageenan quality, and disease resistance have been observed to be gradually declining. These changes and challenges are also exacerbated by global climate change. Over the last decade, there is a global increase in demand in various industries for carrageenan. It is therefore imperative that studies focusing on selecting new superior strains with high carrageenan yield and quality satisfy the demands of various processing industries. These objectives can be achieved through the establishment and maintenance of seaweed seed banks. Here at the University of the Philippines, the in vitro gene bank was established at the Marine Science Institute, the Seaweed Culture Laboratory, and Gene Bank. The Gene Bank was originally established in year 2000 to house branch cultures of farmed or cultivated strains in the Philippines. Oldest collections date back as early as 2001, 2002, 2006, and 2009. Branch cultures of Tambalang and Giant from Zamboanga, a local variety of Capaphycos alvarezii, are more than 20 years old. Some of the common commercially farmed varieties housed in the gene bank are Tambalang, Giant, and Sakol. Our studies indicated a rich reservoir of unutilized wild genotypes in the Philippines that can be used in developing new cultivars with superior traits. UPMSI Seaweed Culture Laboratory and Gene Bank is the only institute in the eukimatoid farming countries in the tropics that is holding more than 500 unique strains or cultivars of eukimatoids that will be further expanded. Through the support of Global Seaweed Coalition funded Secure Future project, most of these have been genotyped and phenotypic characterization is continuing. DNA barcodes generated from our studies effectively expand the existing databank of Capaphycos sequences and can provide insights in elucidating the genetic diversity of Capaphycos species in the country as well as neighboring countries of the Coral Triangle. In addition, we were also able to produce progeny from novel strains of Capaphycos alvarezii through in vitro spore germination from tetrasporic and carposporic individuals from the wild. The seed bank currently have more than 90 culture jars of sporelings produced from different parental plants. Culture handling and cleaning is an important and crucial aspect in maintaining the culture. These videos show how our culture stewards meticulously clean and gently handle the cultures with tender, loving care. Aside from the weekly cleaning of the cultures, we also prepare culture media, autoclave the seawater, and sterilize the glasswares before use. While the gene bank is expanding, one challenge is maintaining a steady supply of seawater, which we currently source from our marine station in Bolinao, Pangasinan, which is a 7-hour drive from UP Marine Science Institute. Voucher specimens of the eukimatoid collections were also done and recorded in the database. Complementary to the in vitro gene bank, field or hatchery gene banks are best suited for the conservation of clonal crops. The UP Marine Science Institute Outdoor Land-Based Nursery, or OLBN, is situated in our marine station at the Bolinao Marine Laboratory in the coastal town of Bolinao, Pangasinan. A field or hatchery gene bank provides easy and ready access to the crop's genetic resources under continuous characterization, evaluation of their phenotypic traits such as growth, chemistry, and susceptibility to disease, among others, and their immediate utilization while the same material is conserved in vitro gene bank in the form of branch cuttings. The outdoor land-based nursery houses collections from the wild, commercial strains from different farm localities in the Philippines, as well as tetrasporophyte and gametophyte individuals generated from spores. We are currently maintaining 124 glass tanks that contains a total of 98 unique individuals. Here, we showcase some of the commercial and novel strains that exhibit unique morphological expressions. Aside from the glass tank setup, we also maintain concrete tanks or raceways. The raceways are important for faster production of biomass, which will be utilized for in situ studies or distribution to farmers for test planting. Consistent monitoring of plant health is important to prevent disease outbreaks or epiphyte infestation. Our hatchery stewards not only monitor plant health but also see to it that the hatchery water supply, filtration, and aeration systems are properly working and are in good condition. Ambient light, temperature, and salinity are also monitored and recorded. Under hatchery conditions, we have recorded growth rates of the novel strains that range from 3 to 6%. These values are relatively comparable to the commercial strains that we have. After which, these fast-growing strains were further tested for carrageenan analysis. Moreover, through the field gene bank, we were able to conduct detailed phenotypic studies on growth and chemistry, such as carbohydrate and protein contents. However, more efforts will be done to assess the biochemical characteristics of different strains. In situ experiments were also conducted to test the growth performance of the different strains in the sea. This is a joint effort with the local government unit, the Bureau of Fisheries and Aquatic Resources, and our partner NGO, Coast 4C. In addition to these efforts, different culture techniques were also explored, including methods such as micropropagation and application of biostimulants. Plant health is vital, and the field hatchery gene banks can be particularly sensitive to health issues such that regular monitoring and testing, coupled with application of disease control measures, is essential to maintain plants free of pests. It is therefore important to study and characterize the diseases we observe that plagues our plant cultures. Careful planning and field management are necessary to mitigate these biotic challenges. The only suitable techniques to preserve eukymatoid cultivars is through in vitro banking and field gene banking. These facilities should remain freely accessible to breeders, allowing them to work on increasing production yield, disease resistance, environmental stress tolerance, and the chemical or nutritional qualities of these economically important seaweeds. With this, we thank the Global Seaweed Coalition for the continued support in these initiatives.